My name is Randy Sheckman from UC-Berkeley. I've been asked to make introductions of this year's winners. And I will do so, but I was only given 10 minutes, but I'm going to engage in a bit of civil disobedience here because I think the winners deserve more recognition than 10 minutes would afford for 6 wonderful people. So first, and as I call your name, would you please come up and take a seat at the front. David Allis, professor and head of the Laboratory of Chromatin Biology and Epigenetics at Rockefeller University. Chromatin is the physiologic template of our genome. The packaging of have a seat. This is going to take a while. The packaging of DNA within chromatin, the orderly replication and distribution of chromosomes, the maintenance of genome integrity, and the regulated expression of genes depend upon the highly conserved histone proteins. Despite a long-standing appreciation of the primary structure of histones, various covalent modifications and speculation about regulatory roles for histones in gene expression, the field was plagued for many years because of poor methods for histone chromatin isolation, consequent histone chromatin aggregation, and lack of any clear distinction between a basic uniform chromatin structure versus a more heterogeneous array of histone modifications along the genome. It was generally believed that histone proteins were passive participants in packaging DNA into a more manageable form. In pursuit of understanding of the DNA histone protein complex and the intricate system that allows for gene expression, Allis's lab focused on chromatin signaling via histone modifications, acetylation, methylation, and phosphorylation. Before Allis's work, it was not appreciated that histone proteins might play an active role in dictating meaningful biological responses. No histone modifying activity was known. Thus, there was no reason to anticipate that transcription machinery might possess histone-modifying enzymatic functions. Allis is best known for deciphering regulatory mechanisms that impinge on the fundamental repeating unit of chromatin and for identifying the responsible enzyme systems that govern the covalent modifications of the histone proteins, the principal components that organize chromatin. Allis discovered the critical link through histone acetyltransferase containing transcriptional coactivators between targeted histone acetylation and gene-specific transcription activation. In further studies, he linked histone phosphorylation events to mitosis and mitogen action, established a synergy between histone phosphorylation and acetylation events, and elaborated the histone code hypothesis, one of the most highly cited theories governing epigenetics. Implications of this research for human biology and human disease, notably cancer, are far-reaching and continue at a remarkable pace. Next, I'd like to call forward Benabid, Dr. Benabid. Dr. Benabid, along with his collaborator and last year's Breakthrough Prize winner, daylong, devised an effective and reversible intervention that remedies neuronal misfiring. Their work has culminated in an effective treatment for more than 100,000 individuals worldwide with severe illness who suffer from complications of Parkinson's disease. Parkinson's disease is commonly treated with medicines such as levodopa to improve muscle control, balance, and walking. But higher doses tend to be needed over time in this progressive neurological condition, and long-term use can lead to motor fluctuations such as tremor, rigidity, or slowness. Prior to the development of deep brain stimulation, or DBS, the main surgical treatment of Parkinson's disease was lesioning to inhibit involuntary abnormal motion associated with severe Parkinson's not controlled by optimal medication. In 1987, Benabid was about to create a lesion in a person with an essential tremor, a condition that causes trembling in various parts of the body. He was targeting a component of the thalamus that contributes to tremors. As usual, the patient was awake, so Benabid could test whether he had located the right tissue. He inserted a probe into the spot that he intended to lesion and sent an electrical pulse to ensure that perturbing this site did not generate undesired effects. Usually he delivered 50 hertz, but he decided to find out what would happen if he increased the frequency to a higher level. Just below 100 hertz, something unexpected happened. The tremor stopped. The patient became so still, Benabid thought that he'd caused an unintended muscle contraction. He switched off the stimulation and apologized for his mistake. The patient told him not to apologize, as it was the first time in many years that he hadn't shaken. Benabid repeated the procedure with the same outcome. Furthermore, when he withdrew the current, the tremor recurred. The effect, therefore, was reversible. Benabid realized that he was onto something exciting. Deep brain stimulation had been used for more than 2 decades to treat pain, but no one had dialed up the frequency. Later that year, he tried the approach for Parkinson patients. In addition, he implanted a device that was on the market for pain relief and delivers constant stimulation. Some of the individuals benefited from the procedure and no complications occurred. In 1991, Benabid reported that high-frequency stimulation could be deployed bilaterally in people with essential tremor and PD. The strategy reduced tremor on both sides of the body. The gains were long-lasting, and the adverse effects were mild. Furthermore, any desired outcomes could be reversed by reducing stimulation. Later animal studies indicated that the subthalamic nucleus might be more effective as a target. As technology advanced to allow such stimulation to be continuously applied for a long time. DBS became widely adopted in the 1990s for treatment of movement disorders in Parkinson's. In 2002, the U.S. Food and Drug Administration approved high-frequency stimulation of the subthalamic nucleus for treating advanced Parkinson's. And let me add on a personal note that I'm grateful to Dr. Benabid because my wife suffers from Parkinson and had this procedure 5 years ago. And although she has advanced dementia, the movement aspects of the disease have been completely eliminated. Next, let me introduce Emmanuel Charpentier and Jennifer Doudna. Please come forward together. Jennifer Doudna, Emmanuel is an Alexander von Humboldt professor at Hanover Medical School and the Helmholtz Center for Infection Research, as well as a guest professor in UMIA at UMIA University. And Jennifer is my colleague in the Department of Molecular and Cell Biology and a Howard Hughes investigator at UC-Berkeley. To protect themselves from attack by invading alien genomes, phages, plasmids, bacteria and archaea have evolved an RNA-guided adaptive immunity system called CRISPR-Cas, short for clustered, regularly interspersed short palindromic repeats, CRISPR-associated proteins. Around 2005, some started to suspect that CRISPR was part of a bacterial defense against external aggression. Shortly thereafter, it became clear that it actually was an adaptive immune system in bacteria. In other words, a defense with the ability to remember the invading virus and thus be better equipped against future attacks. The concept that bacteria could be adaptive, have adaptive immune systems, was previously unknown. Charpentier's lab discovered that this small tracker RNA molecule was involved in a fundamentally novel mechanism of activation of one of the CRISPR systems, the CRISPR-Cas9 system. She decided to follow up with an analysis of the molecular mechanisms involved, showing in detail how CRISPR neutralizes a virus that has gotten into bacterial DNA. Doudna made outstanding contributions to understanding how non-coding RNAs function in living systems. Following her pioneering work on the structures and catalytic mechanisms of ribozymes, Doudna made exceptional contributions to understanding how small RNAs are produced and used to control gene expression in mammals and bacteria. Together, Doudna and Charpentier and their colleagues determined that Cas9, an enzyme specialized for cutting DNA, can be programmed with a single RNA molecule to cleave specific DNA sites, creating a simple and versatile system for genome targeting and editing. Together, they published, Jennifer published a very important paper in 2010. Following on the initial observation, she, through structural analysis, helped identify the sequence-specific and structure-specific processing of RNA by the large family of CRISPR endonucleases. And together, they collaborated, showing that the system was programmable by changing the guide RNA, and therefore, it had the potential for genome editing. Doudna's research further showed how the system could be readily adapted for use in human and other cells and organisms. This transformative technology has influenced the entire field of biology by enabling experiments that were never simple before to conduct or even possible to conceive. So congratulations to both of you. Finally then, let me introduce Victor Ambros and Gary Ruvkun. Please come forward. Victor is a professor of molecular medicine at UMass Medical School, and Gary is at the Mass General Hospital, Harvard Medical School. Welcome on stage. They are credited for their pioneering discoveries of the existence and function of microRNAs and small interfering RNAs, molecules that are now known to play a critical role in gene expression. The road to discovery of microRNAs and their function began in the 1980s when Ambros and Ruvkun were postdoctoral fellows in Bob Horvitz's lab at MIT. They were studying the Lin4 gene and Lin14 genes that regulate developmental timing in the nematode C. elegans. Mutation of Lin4 kept the worm's larvae from developing into fully formed animals, while mutations in another gene, Lin14, caused the larvae to mature prematurely. Ambros and Ruvkun collaborated on the isolation of the Lin14 gene. In 1989, Ambros, in his own lab at Harvard, established that Lin4 acts as a repressor of Lin14 activity, although the molecular mechanism for that repression awaited further investigation. In 91, Ruvkun and his colleagues at Harvard established that genetic anomalies in Lin14 sequence, specifically in an area of the gene called the 3' untranslated, or UTR, were associated with excess production of the Lin14 protein produced from the messenger RNA that Lin4 targets. A year later, Ambros and his colleagues successfully isolated and cloned Lin4. To his surprise, Ambros also found that the gene's product was not a standard regulatory protein, but a tiny non-coding strand of RNA, about 22 nucleotides long, that is conserved in other nematode species, showing a selection on its RNA sequence. At around the same time, Ruvkun demonstrated that particular elements of the Lin14 3' UTR are conserved in other nematodes, suggesting evolutionary selection on their function. Working together, Ambros and Ruvkun compared the Lin4 and Lin14 sequences and discovered that the 22 nucleotide Lin4 RNA and the 3' UTR were partially complementary, and that the short regions of complementarity were highly conserved in evolutionary comparisons to other nematode Lin4 and 14 genes. They hypothesized that this RNA regulated the gene by binding to the 3' UTR. And Ruvkun showed that repression mediated by this was via control at the level of translation. Together, they published in 1993 remarkable findings. But the broader importance of these findings, the idea that small RNAs, later dubbed microRNAs, might play a role in gene expression beyond C. elegans, was not immediately clear. This was followed then by Ruvkun's twin discoveries of a second microRNA, and subsequently, as you know, many, many more. In the ensuing years, the study of these and other related classes of small RNAs has exploded into an exciting new field of research. Scientists have linked the gene silencing abilities of these tiny molecules to a diverse range of important developmental and physiologic processes in both plants and animals. And so I congratulate you both and all of you for your achievements. Now I think we'll move on with Ron Vale. Where are you, Ron? Okay.
I'm Ron Vale from UCSF, and first of all, I'd like to extend my congratulations to all the prize winners. It's fantastic. As my job, I was assigned to moderate a panel discussion that would be of interest with this group for the young scientists in the audience. So I figured with that job, the best thing I could do is step back and actually let young scientists organize this panel discussion. So this group of students and postdocs here I'll introduce have actually been talking to one another via Skype and Google Docs to create a set of questions for the panelists here. So just to introduce them, Angelica Parente here. Angelica. Louise Guillaume, yeah, say hello. Livy Wills from Berkeley, I should say. Last two are from Stanford. Sam Sternberg from Berkeley. And Flora Rutaganira, that's right, from UCSF, and Adam Williamson from UCSF too. So I think in general they have one question at the beginning for all of you, another question at the end for all of you, and in between they've organized questions that they're going to ask either pairs of you or individually. So with that, I'm going to let them take the floor here. Flora?
So for this question, it's going to be directed to each of you on the panel. And if you could keep your answers brief, we would appreciate it. So we're going to start off with an easy-ish question. Can you please tell us about one pivotal person, such as a mentor or a hero, or an experience that profoundly influenced your early scientific career? And if we could start with David, Allis, Dr. Allis, that'd be awesome. Thank you.
Yep. Thank you very much. And, I'm honored to sit up here with the others and congratulate everyone from past years and thank all the sponsoring institutions. So my quick answer would be that's an easy question for me because I would sort of put the life-changing experience for me in terms of my career with my PhD advisor and my subsequent postdoc advisor, who both, I think, developed a lab sort of environment that was just a lot of fun to work in. So I never actually thought of a lab as not being a lot of fun past the science alone in terms of, great lab mates. So I thought if I ever could cut it and never make it to the end of the road and have my own lab, I sort of wanted to copy that. If we could do great science or good science, but do it where we actually had fun together, I thought that would be the best sort of that would be the best formula for ultimate success.
Wait, you got to name names.
Yeah. So my PhD advisor was Tony Mahowald, who's now retired, and my postdoc advisor was Marty Gorofsky. Thank you. Those are the names.
So my name is Alim Louis Benabid. I'm from France. I'm basically a neurosurgeon, and I've got similar, at the same time, a PhD in physics, which has been helpful for the rest of my career. My career has been particularly specific because it was luck and luck and luck and luck and luck all along the career. I wish you the same. Second, which is not necessarily a good example in the States, I was born in the hospital in Grenoble. I did all my training in the hospital in Grenoble. I had all my career in the hospital of Grenoble. I did my retirement close to the hospital of Grenoble. And the only thing in my will is I don't want to be buried in the hospital cemetery. Then, to be serious, I had many, many people helping me all along. The list would be too long. Two people particularly were important. One was a professor of biochemistry. When I got my professorship quite early, by chance, of course, by luck, he advised me. I said, what should I do now? He said, go away. I said, well, my family has been waiting for long that I had a stabilized position. He said, go away. You have to learn outside. And I went to San Diego at the Salk Institute, and I spent 1 year with the second person to whom I owe a lot, who is Floyd Bloom, who taught me very briefly that I didn't know anything in the brain, and it was time for me to start, so which I did during my stay, which is one very important period of my life, because it changed really the way I had planned it before. So this is essentially what I want to say. And if I return to my staying permanently in Grenoble, this is not the way that it happens always in France. Normally people move. They don't move as often as you do here, because sometimes when someone moves, the suspicion is that he has been fired, so nobody wants him to come. But to show also that in this situation at least, but in other parts of the world, it is possible to make a good career being in the same place, which does not prevent you to move and have different stays and stages, which I did a lot, and I traveled a lot. So this is, I hope I have.
Hi, well, this is I really have to, first of all, thank the students and Ron for organizing this really interesting forum. And for me, I guess I would say there really are 4 people that were critical to me at different stages in my career. The first is someone I don't know the name of. You know, I'm kind of embarrassed that I don't know her name, but this was something that happened when I was a junior in high school. I was at a big public high school in Hawaii, and not a particularly great school, but I knew I loved science, I knew I loved math. Nobody in my family was a scientist, so never had occurred to me that I could, you know, be a scientist. And then one day a woman came to our school to talk about her research, and she was from the Cancer Center on Oahu in Honolulu, and she talked about her work as a biochemist studying cancer cells and how they become defective. And I was so captivated that I just thought, I want to do that. I knew I wanted to do it. It was just almost a moment, you know, just saying, I know that's what I really want to do in my life. And so then I went to college, Pomona College in California, and I went there because they had a biochemistry major. This was in the early 80s. Not many undergraduate programs had biochemistry majors in those days, but Pomona did. And I had the good fortune to work in the lab of Sharon Panasenko, who was a newly hired biochemistry professor at Pomona. And she gave me the opportunity to work in her lab. I was not the top student in her biochemistry class, but she still selected me to come and work in her lab over the summer. And it was just a spectacular, transforming experience for me. I loved it, and I knew that was the type of work that I wanted to do in the future. So then I went on to graduate school. I went to Harvard Medical School for grad school. And again, sort of a serendipitous thing, I ended up doing a rotation in the lab of Jack Shostak, who at the time was a yeast geneticist, and I had been a chemistry major in college, so I didn't really know anything about genetics, really nothing. But again, he gave me a chance to work in his lab, and he was so creative and encouraging and supportive, and just, you know, really built up my confidence in myself that I really could do this kind of science, that it was, you know, it was just, again, a really important time in my career to give me that sort of support. And then finally, I ended up going to Colorado to do a postdoctoral fellowship, and I actually had gone there to work with a different person, but I ended up, for various reasons, in the lab of Tom Check. And Tom, again, was just an incredibly supportive mentor who encouraged me to pursue my interests. I was, at the time, doing structural biology on RNA, something that was very new, and many people were advising me that it would never work, and he, you know, was supportive of me and gave me the freedom to pursue the things that I was really excited about doing. So I think, you know, it just sort of underscores the importance of having people that believe in you and you know, give you support at critical times in your career. And I would say for the students that, you know, you look for those opportunities to find people that are going to give you that kind of support and advice and help you pursue your passions, whatever they might be.
Okay, so me, I started actually my career at the Pasteur Institute in the laboratory of Patrice Courvalin, who's a big name in the antibiotic resistance field in the world. And I should say that, yes, actually it's really at the level of my master. When I entered the lab, that I figured out that it was really the place where I would like to stay. I just felt home. And I think this was important for me. And so it's really at this moment that I felt, okay, this is what fits to me. Also, what I think I appreciated at the time is the understanding very early on that it's a multidisciplinary job, if one can call it a job, where you can really fulfill a lot of aspects of your curiosity, of quest for knowledge and understanding, where you have the possibility to, at the end of the career, when you're a leader, where you can combine different skills of being a manager, thinking of your science, working with very young, talented scientists, collaborating, traveling. And actually, this is really when I arrived at the Rockefeller University in New York, that I had a little bit another perspective on what was to be a scientist. Just because it's maybe a little bit a different way of doing science. So I was arriving at the Rockefeller as a postdoc, and I had maybe more the freedom to choose a little bit the path that I wanted to take. So actually I did 3 postdocs in New York, and I changed quite much field of research. This was always a reproach. But at the end of the day, it paid off a lot because I think I got to really enter different topics and have more a larger view on biology. And also what was very nice with this strategies that I had is that when I built up my lab in Austria after my time in the US, I figured out that maybe I was a little bit too curious, and because I was starting with too many projects. But in principle, it allowed me to approach different questions, knowing that I had a question and I will be able to follow up on the mechanistics and the pathways for the different questions. And that if really there was something I was not really that specialized of, I will be able to collaborate with colleagues. But I think it's I don't know, for my case, it has been nice to have diversity in my research, just to be a little bit more open to different questions and how to approach questions. And
This is a kind of question that I could go on and on and on about and give you all kinds of different examples of throughout life where something happened that had not happened, I wouldn't be here, And obviously, my wife, Rosalind Lee, is incredibly centrally important to my career. And I won't go on and on about this because I don't want to embarrass her too much. She's in the room. But many of you may know that, she's the first author on one of the most important, one of the papers that Randy was telling you about. And she and Rhonda Feinbaum did the work, that we're being noted for. So, it's clearly, and, she's been working with me for a long, long time. So, if you delete Candy, you essentially delete me too. So, from the scene. But one thing I wanted to bring up, which was very important to me, is I landed at MIT extremely naive. Essentially, I was an idiot, and believe it or not, the MIT environment was welcoming, and so you found out instantly that you were not the top of the heap. In fact, you were lucky if you were class average, and that's a good day, And so, for some reason, I was very happy to find out that not everybody has to be like incredibly smart. And here I am, and they still haven't thrown me out yet. And moreover, they let me work in a lab, and I eventually ended up working for Jerry Letvin, believe it or not, if some of you know him, and then David Baltimore. And so here were people who were super, super world-class folks, and they were letting me work in their lab. And all the people around me were amazing, lots of smart people. And so I became, comfortable with the idea that I can fit into the scientific community and make a contribution. These folks are really smart, but they still let me work with them, And now I understand, as a PI, the way it works is you need to have that mixture of folks in your lab. So yeah, that's what I had to say.
So sort of calculation, at one point I was trying to figure out how important I am to my lab. These things go to your head, you know. And so my lab is about 500 person years of people who've worked in it over the time. And I account for 30 or 35 of those person years. So the culture of the lab, the most I can claim, let's say I inflate myself twofold, you know, it's still only 10% of the lab, right? And so the culture has a culture of its own, and that's true of every lab. And so the mentorship is really a mentorship of your peers, and the most I can ascribe to the famous people who might populate each institution is sort of a 2 or 3-fold, you know, per capita brilliance quotient or something, you know, but it's still, the culture is embodied in the people who are in each lab, in each department. And what you really remember over time, you know, the inspiration is from your peers. I'm still, you know, best friends with the people I was in graduate school with. We still sort of keep each other apprised of what we're doing and still intense about science. And it's a wonderful thing to have relationships over 40 years that are sort of, where a focus, a major focus is scientific intensity.
Okay, I'm going to direct the next question to Professors Charpentier and Ambros. So it's been an inspiration to me that 4 of this year's Breakthrough Prize winners are being awarded for discoveries that were really the result of a true team effort between 2 independent laboratories. So I'd like you to comment on how we as scientists can follow your example and maintain this kind of open, collaborative atmosphere during a time when we're, of course, competing for the same prizes and especially for some of the same dwindling funding sources. Maybe we can start for Professor Ambros.
Yeah, we haven't practiced joint answers, so this is not fair.
It doesn't have to be joint. You can
Yeah, right.
go separately.
Yeah, you know, that's really so important, what you pointed out. You know, and with, you know, this, as Randy outlined, Gary's and my lab shared data, and we made a discovery together. And as you know, because we actually found that the sequences for complementary, so it's almost like it's sort of you know, wonderful. It's emblematic, so to speak, for how what people can be doing in different labs can be complementary. And if you know, what I've come to learn over the years is that the more the communication, the better. You know, and you share ideas, they multiply. If you think you have an idea that nobody else had and you keep it to yourself, it's been had for sure. Somebody else has that idea. So that idea, that illusion that you somehow have discovered something that nobody else has ever thought of, and you have to keep it and kind of then publish it and scoop people. I think it's just that's a sad trap to be into. And in the last 7 years or so, I've been at UMass Worcester, which is an extremely extraordinary environment that is where people are promiscuous with ideas. I mean, it's almost, you know, embarrassing how people will just spew ideas, you know, a minute by minute, you know, in casual conversation. So it's the most fun I've ever had in science is in the environment that is the most sharing of ideas. So, you know, I would say that when in doubt, give it up and talk about it. You know.
I totally agree with what Victor said, and I think in our days when you want to really understand a pathway, a molecular mechanism, I mean, you know it well, so you need to use a multidisciplinary approach if you want to combine the biology with really the deep understanding at the molecular level. And I think in biology you often combine structural biology with biochemistry, with molecular biology, with cell biology, with genetics. And I think if you really want to make sure that you go in the right way and that you can really understand the phenomenon with different angles, I think it is very important to collaborate and to collaborate early on, because you will be able to understand better the specific pathway or mechanism. And I think it's really beyond this. I think for me it's more the fun of really sharing knowledge and ideas with other types of scientists who may have another angle of thoughts. And I think it's, we are living in a world of communication, and I just realized, because also I have 2 labs, that the young people in my lab, they really like to work with people on the other side of the Atlantic Ocean. I think it's very much stimulating. And communicating via Skype, it's kind of exotic. And the idea that, okay, you have a buddy on the other side of the Atlantic Ocean who's working together with you to really pursue a biological phenomenon, I think it's very stimulating. And I think this is for me, because I traveled a lot in Europe as well, this is really for me the nice part of science. I mean, it's a lot about meeting scientists, communicating the science, trying to have fun in science, to also be confronted to different cultures. And I think the collaboration is really part of it. Mm-hmm.
Thank you.
So this question is directed at Dr. Benabid and Professor Doudna. The Breakthrough Prize is awarded for discoveries that extend human life, but these will most immediately be available only to the privileged. Given that biotechnology is guiding the way from relatively low-cost treatments over usually the course of someone's entire life to high-cost cures that they would only have to take once, how do we balance our excitement over these discoveries with the consideration of developing parts of the world in need of basic medicine?
Well, I think when you are trying to design a new method for treatment, either chemical or technological, you don't have to talk about money, because it costs extremely highly, and only the results count. And if the treatment is valuable, if it helps people, then there is always a way to go around. And typically, deep brain stimulators, which I use, if you do them on the bench in the lab or in an associated lab, it costs millions. And then you buy it for several 10,000 euros or dollars. Same for drugs. They are extremely expensive for the company to develop them. And then after, you have to find a price which is affordable for anyone, including the funding agencies. This is a problem, this is true, and I think that I take advantage of your question to say that if we are designing a new method, we have to keep in mind that at a later stage, they have to be available for everyone, I would say, on Earth. A method which is restricted to a happy few is not a method, and not a valid method. So this is what technology helps today. The costs are going down, whatever we think, and there are, just to talk about the silicon chips, etc., the price of today compared to the price of 20 years ago is not comparable. What was particular in my approach of treatment, because I was a doctor, particularly a surgeon, it has to be a translational research. I don't like the difference between fundamental and applied. There is always a moment where a fundamental research becomes applied, and there is always a moment where applied methods need some fundamental approach. And in the situation where I work now, which is after my retirement, I've been picked up by what is called the French Atomic Energy Agency, which has nothing to do with atom, actually, but that's a good thing because the cost of nuclear things is transformed in funding for research. We have, I would say, almost unlimited funding for once the program are accepted. And I'm working on several programs currently. One is going beyond deep brain stimulation to cure the disease instead of just taking care of the symptoms. This is infrared illumination, which is just starting this year and very promising. And then we are designing an implant which delivers laser radiation into the brain continuously. The other part is to make tetraplegic patients able to walk by designing an exoskeleton, motorized exoskeleton, and taking the information by implants on the cortex. This last program has costed so far about 35 million euros, and it should be available next year. Doesn't mean that we will have to pay that amount. And the other one also is costly for the moment, but if they are working, they will find the funding even from the funding agencies in the countries. Is that 1 minute?
So I would say that the CRISPR-Cas technology is at a much earlier stage than your work on deep brain stimulation. It's not yet being used in patients. I think one of the things that's very exciting about the technology is that it's easy to use and it's simple to use, right? So there are other technologies for genome engineering, but they never took off the way this one did because they were expensive and for various reasons kind of challenging for labs to get going on. And so I think what I anticipate in this field is that we'll see this technology being used fairly quickly to do things that will help many people very widely and not necessarily as a direct therapeutic, but for doing things like drug discovery research and validating drug targets in human cell lines, making it possible to generate animal models of human disease much more easily and that sort of thing. And so I think the exciting thing there is that will actually impact many people very, very broadly. And then going forward, obviously there's lots of interest in developing the CRISPR-Cas system for human therapeutic applications. And there, it's going to be a longer path just necessarily in terms of, getting through all the various hurdles that's going to require. And it's a bit early to know, I think, really how, what are going to be the most impactful and earliest uses of the system for therapeutic applications. So I would say, stay tuned. But it's a very interesting question, something I think a lot about, and we'll be very interested to kind of stay on top of the field as things go forward and see how we can contribute to making the system better, easier to use, accessible widely to labs across the world, that sort of thing.
The next question is directed to Professors Allis and Ruvkun. The Breakthrough Prize is a generous award from a private foundation. What is the current role of private funding, and where do you see its role changing in the coming years?
Well, certainly, I would have never, ever in a million years predicted that I'd be sitting up on this stage talking about breakthrough prizes. When I was in your stage, every experiment I did, something went wrong. And I'm positive I said, give me a break. But I never said, give me a breakthrough. But more seriously, I think, money is tight. And so it came up this morning, what can the top administrators do and beyond to sort of help mechanistically generate support. And everything has its time and place. And in my own particular research now, epigenetics has become a craze. And Rockefeller University, where I'm at, it's a small campus. But across the street, I'm really blessed. Memorial Sloan Kettering, big cancer hospital. Right up the street, Weill Cornell. And now there's been initiatives in the city, this tri-institutional area, to go to, I want to say, non-federal, I want to say donors of various types with various, ideas in mind, including now cancer, epigenetic therapies that have proven useful in humans. So I support that all the way. If I'm a poster child for them, because anybody that knows my career knows we study the most fundamental mechanisms with the hope that if we can figure out the basic mechanisms, then we can come up later with the therapies and the treatments. So if I'm a poster child for that kind of program and that kind of initiative, that's maybe my role. So I'm a good cheerleader. So I'll defer to Gary.
Yeah, there's many answers. I think the NIH funding of science has not been as good at inspiring the public as NASA, for example. And so we have a lot to learn just from watching how NASA does public outreach. They've been in the business of inspiring kids and other countries because it's all about the world looking up to the U.S., what the space program was about. And so it's almost a State Department issue. And the U.S. should grab onto more credit for what we've done. The genome initiatives were, driven. I know it's not only the U.S., but it was dominated by the U.S. BLAST initiatives, all of that are a gift to the world, and we don't trumpet that very much. And so there's more to be done for sort of selling that. I also think my generation of scientists have been doing a disservice to the next generation because at least me, I'm a whiner, right? I'm just constantly moaning about the grants and the papers and all the tourists of science, right? And my mentors didn't do that. First of all, it was a little easier because they were all like walking on water, but also they just didn't moan as much, and so I think we're complaining way too much for our students because it really ain't that bad, but we're just whiners, yeah.
Okay, so now we're switching to some targeted questions to individual prize winners. And first, I have a question for Professor Doudna. Other than your own work and genomic editing, what areas of research are you most excited about for the future?
Well, I think we heard some discussion this morning about where are the next breakthroughs going to occur, and I really would concur with the field of neurobiology. I think it's very, very exciting times. Back when I was a graduate student and trying to think about what was I going to do for a postdoc, my advisor, Jack Shostak, sat me down and said, I really think you should get into brain research, think about neurobiology. And that was probably a very, very, very wise advice that I didn't take. But I think it's an exciting time because I think that you can see a lot of things coming together that are going to make it possible, I think, to for really big discoveries to be made in that area in the not too distant future. And I think the other thing that I'm very excited about right now in science is that I think that what I've seen over my career, so I've now been running my independent research lab for 20 years, and I've seen a very interesting thing happen in the microcosm of my own lab, and I feel it's true across certainly the biological sciences in general, is that over the last maybe 5 or 6 years, our lab has become much more collaborative, and many if not most of the projects we're working on involve multidisciplinary efforts, and not just with other biologists, but increasingly with people in statistics, mathematics, physics, certainly chemistry, et cetera. And so I think that this is something that we're going to see happening more and more, is these collaborative efforts that bring together different disciplines to address specific problems.
Great, thank you. This question is for Professor Ruvkun. What's your mentorship philosophy? And do you favor hiring talented people and giving them free rein, or do you take an active role in the development of both students and postdocs in your group?
You found my vulnerable spot. Damn. Oh, Jesus. Is this being recorded? So I'm not much of a mentor in terms of, sort of micromanaging what lane should be on what gel, and I'm not very good at controls and things like that. So it's a different kind of mentorship than that. I've heard people in my lab refer to me as the least grown-up person in the lab. And I think it's fairly accurate. So maybe I should just repeat what Charles Barkley said, I ain't no damn role model. So my lab sort of runs in its own way. I don't think it's going to be typical of other labs, but people have lots of freedom so a couple good things. I never beat up on people. Yeah, 1 minute. Yeah, you're right. So I never tell people you've got to be better than you are or something like that. And I think there's a whole sort of Marine Corps mentality, I don't know, in every workplace of people getting beaten up by their. Dilbert has done a good job of taking care of that, sort of bosses who suck. So I don't have these kinds of, here's what you have to do to improve. It's because I think it all comes from seeing each other. Again, it's coming back to the community of a lab. And so what motivates people in my lab to do well is when the next person gives a group meeting that just knocks your socks off. And it's not like I get up there and go, now, see, that's what you should, no, you don't have to do that. It's obvious.
Thank you. So the next question is directed to Dr. Allis. So you had many struggles as an early career scientist, as I read in PNAS, studying chromatin, a not so popular topic at the time, and even worse in a ciliated protozoan. Did you at any time want to jump ship? And how did your early scientific experience shape your approach to mentorship?
I would say absolutely. You couldn't maybe pick a worse career path. I wanted to be an MD, and then that didn't pan out. That was a total disappointment to my family. And then somebody said, you ever tried a lab? And then that ultimately took me to Drosophila, which also wasn't cool, but it was cooler than ciliated protozoan. But in my heart, I was interested in chromatin biology. I thought that I would have to pick a model organism where the sort of active versus inactive components of the nuclear chromatin weren't all mixed together. And thankfully, a former lab postdoc from my PhD lab said, have you ever heard about tetrahymena? And I was like, I don't even know how to spell tetrahymena. But he said, no, that's a fantastic, simple organism. And we've heard a lot about it today. Comments were made about telomeres and telomerase. Tom Check's name came up this morning. So, I really thought this was the perfect organism where you can separate the genomes. Marty Grofsky, with whom I postdoced, he'd already learned to separate the two nuclei, and for me, the rest sort of was magical. But, of course, getting funding and starting your own lab and your first R01, I felt like it's probably game over. Oh, running out of time, we're getting a signal. So, and then not to mention names, but a very good West Coast lab deleted the histone tails genetically in yeast and published a high visibility cell paper that said that these histone domains were dispensable. And I remember my chair at the time, I was at Baylor College of Medicine, was a lipid biochemist. He pulled me into his office and had the cell volume. He said, I thought you told us these proteins were important. It's like, it's over. I went home, I said, it's over. I gave it my best shot. But, we survived that grant round, and I'm grateful, very grateful that we pulled out these enzymes, and,
Professor Allis, once again,
very magical.
Could you please name some names?
The West Coast lab? Is that what you mean?
I'm out of time. I'd like to tell you those names. So yeah, I was a borderline.
So we are running out of time, so this will be the last question. next to last question, sorry. This is directed at Dr. Benabid. There are many neuromyths out there, apps and products promising cognitive improvements based on faulty or embellished scientific evidence. So we can get it straight from a reliable source. Can you realistically foresee any technological advancements that would allow neuromodulation to be used for human enhancement? Will the benefits ever outweigh the risks, and what are the ethical implications?
Thank you for this powerful question. I have 1 minute. I need one more. No, we don't change the mentality of the patients. We are restoring the abnormal behavior to the range of normality. When you have a depressed person and deep brain stimulation makes him not depressed, you have not changed the mentality, as we hear quite often. We have just alleviated the symptoms as we alleviate the tremor. And this is important to consider that. It doesn't mean that by the side effects, for instance, we might be inducing the patient in some behavior which are not the normal ones, like becoming extremely hilariated, etc., etc. So this is a question of adjusting, and that's exactly the same you can do with medication, with philosophies, and even worse, with the politics and the broadcasting. You can make people walking all along same pace. I can't by deep brain stimulation. So, was it your question? I'm not sure. Okay.
So this will be the closing question, which all of you will have a chance to answer, but 1 minute or less.
So first of all, thank you very much for a really fun panel discussion. I think this has been really enlightening for all of us, for sure. So I just wanted to close by having you take a second to think about anyone in particular you'd like to thank as you receive this award. And since we're all thinking about it, if there are any ideas for what you're going to do with your prize money, I think we'd all be curious to hear them. So we'll start with Gary. Thank you very much.
Well, my teachers, Fred Ossibel was this very gentle lab to enter, and he brought the genetic focus of MIT to Harvard. Harvard was always very weak on genetics, surprisingly, and so he brought that. And then going to the Horvitz lab as a postdoc was just an epiphany, and it was an amazing group of people, a sort of magical assembly. And so again, it was the students and postdocs and Bob, all of them sort of transformed me, and I'm very appreciative of that. Yeah, the money I haven't even thought about. Well, a little bit. So it's sort of 20x what a normal professor makes, so, but it's not that every year. So you don't let it, it's not like we're wealthy people now. We just have a big bolus. It's kind of cool.
I mean, it's like an NFL career. It's very short.
So, yeah, right, that's right.
Once again, I want to thank Candy. And I haven't mentioned publicly, my dad, who died just last year, but he was very, very, very important to me because I just remember a vignette would be, like me and my brother would be going to sleep at night, and my dad would come up to tuck us in, and each of us would ask him questions because we know that as long as we were asking him questions, he would hang there by our bed and try to answer the question, And he only had a 9th grade education, so I don't know how much of the answers were ever, actually accurate. But he would do his best to try to answer our questions, and we just tried to dream them up. Like, why is there air? Just to keep him there. And looking back, I realized that kind of like instilled in me a sense of affection for the asking of a question and hearing somebody else kind of talk about it. So that stayed with me. And with regard to what we're going to do with the money, it's been too sudden to really try to plan for us, but we're thinking hard about how to use it as best we can to, help young people get involved in science and stay in science.
Yes, so I can thank my parents, my family, my very close friends, all my colleagues who have always been supportive, my colleagues all around the world. When I was a postdoc and a PhD student, all my friends, students, postdocs, my mentors. All the influential scientists whom I have had the chance to meet even shortly. All the great scientists as well who are not alive, but who have influenced me as well by their wonderful work. And what am I going to do with the money? It's indeed a quite consequent sum of money, especially for a scientist in Europe. I would like to invest part of the money into new ways to think of scientific education, just to try to promote the young generation to choose science for their career. So this is what I would like to do.
So for me, I guess in addition to thanking my mentors, as I already mentioned, there's two people in particular that I wanted to point to. One is my husband, Jamie Kate. So he's been a longtime scientific colleague as well as my life partner and incredible scientist and just a really deep thinker, someone who, we every night at the dinner table, we talk about what we did in the lab today and, we bounce ideas around and we, it's a very, very vibrant kind of relationship that has always really included science front and center. The other person I want to thank is my dad. I liked your comment, Victor. And so my dad has been gone for a long time, but when he was alive, he was not a scientist, but he loved science. And when I was growing up, he would do things like he threw a copy of The Double Helix on my bed one day and said, you really should read this book. And it was it's sort of the first book I read that got me really interested in molecules and how they work in cells and things like that. And then when I was in graduate school, he would come visit me in Colorado and Boulder, and the first night, sit down at the dinner table, and he would say, "So, what's happening in your lab right now?" And he would really want to know the answer. It wasn't just a courtesy question. He wanted to get into it and really talk about the experiments we were doing and why were we doing them and why was this interesting. And it was so incredibly supportive. I always felt really validated by that level of interest. And of course, I want to acknowledge the incredible people I've worked with over the years in the laboratory. Sam, of course, is here, but many other people, Martin Ginnick in particular, who worked on the project that we collaborated on. And this has been, just a real joy for me is to have that level of people in my lab to work with. As far as what I'll do with the money, like the others, it's a bit early. I'm still thinking about it, but I certainly want to find ways to use it to support the science going forward, and in particular to encourage younger scientists to get involved and to know that they will have an opportunity to pursue their passion as I have.
Well, first of all, I would like to thank the founders of the Breakthrough Prize because they have made it, and this is unprecedented. Second, I would like to thank, although they are not here anymore, my parents. My father wanted me to be a doctor. My mother wanted me to be an engineer, so I did both to please them because I love them. I have to, as usual, but it's special, I have to thank my wife. One says that behind a great man there is a great woman. I have an immense woman behind me. She was a scientist, very good scientist also. And all my mentors, my colleagues as a neurosurgeon and a scientist, but in hospital, this is a special relationship with the people working with you. They have to support you. Stop. So a lot of people to thank, and the patients just to trust you. When you think that if you ask someone, be naked, they might be shocked. If you ask, can I put my finger in your brain, they say, please do. I have the feeling that pudeur in French is not always in the right place. What should I do with? as many of us, I never had the time to think of such a money to spend. I could translate the problem to my wife. She would solve it, maybe. I'm sure she won't. I will clearly help my 2 children. I have 2 grown-up children who had difficult life, maybe because I wasn't here, so I will help them. But of course, this has to be also thought in terms of helping. It's not very original, but helping the continuation of research. And at the level where I do my research, I cannot fund the several millions that I need per year, but trying to find the best way to establish kind of modest funding to deliver, let's say, a postdoctorship or something like this, regularly over something longer than my lifetime, because it's not going to be long. Thank you.
I realize the hour is late. Last night we were told to sort of hold our personal comments, but I certainly would have said last night that I'm not only a lab guy, I'm a family guy. And with Seth MacFarlane running the show last night, this gives me at least a chance to certainly thank my wife who's here and my children. They've been very supportive. Of course, my mentors and so on and so forth, as we've heard. I feel really blessed, and largely due to not only their support, but of course all the past lab colleagues, current colleagues, collaborators from all over. I really thrive on that kind of interaction, and so my prize winning here is only a reflection of all their hard work. And in terms of money, we really, I haven't even figured out how many zeros this is, let alone what to do with it. I didn't get the math part as well as I should have. But on a serious note, the thing that my wife and I sort of connect on, she's an elementary school teacher. Now I'm so old that I have grandchildren. And if there's one thing I'm a sucker on, if I'd made it in medicine, I would have liked pediatrics. And so now it's too much science, but what's happened in histone biology is now these high-frequency oncogenic mutations are mapping to pediatric cancers, in particular the most devastating in the brainstem. And so that's a cause that my wife and I think we could connect on, trying to sort of help this DIPG tragedy, try to, in my lab, work on how to detoxify it, but we know this takes money. And so that's a cause that we both really can connect on. And then I'm a big fan of junior scientists. And so if I can find a mechanism to help that generation, like some of you, in some kind of a cool way, that would be fantastic. So that's where I'm at.
Well, hold on. Hold applause to the end. That was an absolutely terrific discussion. I know everyone really enjoyed that. So we thank the prize winners, but I also want to thank the students and the postdocs, Flora, Sam, Livy, Adam, Angelica, and Louise. This was a great inter-institutional collaboration, and I think they put on a really thoughtful set of questions to drive this discussion. So again, just to close, thank you to the prize winners, thank you to the students and postdocs, and now you can applaud for everyone.